Ladies and gentlemen, friends and colleagues, I want to thank the organizers for giving me the opportunity to make this presentation today, and I am truly very sorry that I cannot be with you in beautiful Honolulu. However, I truly appreciate the opportunity to come to you in this audio-visual clip. As you can see from the title of this talk, 3 Decades of HIV, A Personal Journey, I'm going to talk to you about my experiences over the 30 years that I, as an immunologist, has been involved in both the science and the policy of what has turned out to be one of the most transforming pandemics in the history of our civilization. And I'm going to emphasize, importantly, how I came to this as an immunologist. In order to do this, we have to really go back to my earlier training. As many of my friends and colleagues know, I've trained in internal medicine as a classical physician at the New York Hospital Cornell Medical Center, both in medical school and years of internship and residency. I developed an interest in the burgeoning field of human immunology and the interface with infectious diseases. So I wanted to study infectious diseases, but I wanted to do so as an immunologist. In order to do this, I needed special training. I came to the National Institutes of Health, to the National Institutes of Allergy and Infectious Diseases, and I had the fortune to be coupled with a mentor whose interest was host defense mechanisms. Although he was not a classic immunologist, he gave me the opportunity to study fundamental basic immunology in an infectious disease environment. I show you these selected immunology papers, which I published during my 3-year fellowship in a typical immunology approach, studying antibody affinity, at that time what we called antigenic competition, suppression of antibody synthesis, as you can see, a particular interest in the B-cell limb of the immune response. There was a tension between my interest in doing clinical investigation and basic investigation, and so I took an additional year as chief resident in medicine at the New York Hospital Cornell Medical Center. This picture shows me here during that year in 71 to 72. I made the arrangements to come back to the National Institute of Allergy and Infectious Diseases as a senior independent investigator and began doing 2 things. One, I did clinical studies on diseases that were characterized by aberrant immune activation and was able to develop, fortunately, some therapies that turned out to ultimately be either cures or inducers of high levels of remission. However, I continued my intensive work at the bench in immunology. And again, during the period from the time I came back from my chief residency until 1981, I show you some representative papers giving you an idea of the kinds of things I was interested in. The suppression of the immune response in humans by immunosuppressive therapies like glucocorticoids, the study of lymphocyte subpopulations, and always the prevailing interest of the relationship between polyclonal activation and antigenic stimulation, as shown on the bottom of this slide in a JEM paper in 1976. I continued to study over the years the activation and regulation of the human B-cell response. I studied some diseases, again, always characterized by aberrant immune activation, such as vasculitides and systemic lupus erythematosus, and again, always concerned about what role non-specific activation has in specific immune responses. I was happy at this, I was productive at this, but the one gap that was clear to me is that I wanted to move into an area where we can be more related to infectious diseases in general, particularly infectious diseases of some global importance. And then one day, as it happens in many people's career, something happened that completely changed my professional and in many respects, my personal life. It was the first MMWR from the CDC in the summer of 1981, reporting curiously 5 gay men from Los Angeles who were otherwise felt to be healthy, who developed pneumocystis pneumonia, which is only seen in profoundly immunodeficient patients. I didn't make much of it until a month later when the second MMWR came out, again from the CDC, this time in 26, again curiously, only gay men who had not only PCP, but also Kaposi sarcoma, a tumor again seen only in individuals with immune defects. This was beyond Los Angeles and included San Francisco and in New York City. I became profoundly interested in this and made a decision that I was going to pursue this disease despite the fact that I was quite successful in doing what I was doing. This was my laboratory in 1981, the Laboratory of Immunoregulation, a small lab of fellows and students who were interested in studying the regulation of the human immune response in health and disease. We continued our work, but I became even more interested in this syndrome. In fact, I went out on a limb in the winter of 1981 and wrote a paper that was published in the spring of '82. As you can see, the name AIDS was not yet given to this syndrome. And I made a statement that unfortunately turned out to be prescient. I said, because we do not know the cause of this syndrome, any assumption that it will remain restricted to a particular segment of our society is truly an assumption without scientific basis. And as it turned out, I was unfortunately correct. As shown by these sequential MMWRs, it went well beyond the gay population, involving Haitians, hemophiliacs, transfusion recipients, babies born of infected mothers, and female sexual partners of males who had this disease. The only thing we could do was really observational, because at this time, if you look at the dates, the virus had not yet been discovered as the etiologic agent. So I spent my clinical time taking care of individuals with this disease at the same time as I was trying to figure out what an experimental immunological approach would be. And getting back to my lab, here again, circled in red, was an individual who had just recently arrived from medical training at the University of Michigan who wanted to work with me on studying human B cells. I felt this was the opportunity and started our first AIDS group at the NIH, in which we began to study what we did best. And here's a paper from the New England Journal of Medicine called Abnormalities of B cell Activation and Immunoregulation. We made the interesting observation, did not know the significance at the time, that even though this was a disease of profound immunodeficiency, there was aberrant activation of the B cell response in what appeared to be a polyclonal manner, which tickled my interest of years before regarding this phenomenon. We went on and described other abnormalities, including the soluble antigen recognition defect in HIV-infected individuals, and began to intensively study and convert virtually all of my lab to this study. Another very important thing happened in my life at the time. I never felt that I really wanted to do anything that had to do with administration, but I thought that the opportunity of becoming director of the National Institute of Allergy and Infectious Diseases at a time when, in fact, infectious diseases was not nearly as prominent as it really I felt it should be, both domestically and globally, I took the job only under the condition that I would be able to continue to do my basic research in my lab and see patients. I was given the opportunity and I took the job. This opened a whole new world for me. Here I am in one of the, believe it or not, more than 200 testimonies that I made before the United States Congress, either the House or the Senate, talking here at a budget hearing, but talking about a variety of things over many, many years. Another important arena that I entered was that of dealing something that I had never thought that I would have the opportunity to do with various presidents of the United States, all the way from Ronald Reagan through George H.W. Bush, President Clinton, President George W. Bush, and our current president, President Obama. I want to tell you a little bit about this because it does have impact on what evolved over the years with HIV. And that has to do with President George H.W. Bush. I had been very visible in HIV/AIDS as a scientist and in some respects as a policy person because, as I mentioned, President Reagan did not even mention the idea or the word AIDS until the very latter part of his second term. So George W. Bush, the president, came to the NIH as vice president, wanting to run for president, saying he needed to learn something about this new disease, and he'd like to meet with this young immunologist who was studying it. I had the privilege of showing him around for several hours at the NIH, and we developed a strong friendship. In fact, even to this day, he writes me notes when he sees things in the newspaper that relate to what we're doing here at NIH, congratulating us and wishing us well. When he became president, his interest in HIV/AIDS intensified. Here he is visiting one of our wards, and that's me with Sam Broder, with the president and the first lady. I had the opportunity to take him through our wards, and here we are in the solarium in Building 10, where I'm introducing him to several of our HIV-infected individuals. The young man with the vest in the middle between Mrs. Bush and President Bush is one of our patients who articulated some of the trials and tribulations. There was another thing that happened that day that is one of those quirks of fortune that you never know where it's going to lead. In that entourage from the White House was a young person who was a staffer in the White House of his father, George W. Bush, well before he became president of the Texas Rangers and well before he became governor of Texas. I struck up a friendship and a relationship with him that will become very important many years later. We'll get back to that shortly. Let's talk about now fast forwarding to where we are at the present time. 60 million people having been infected, 30 million deaths, 34 million people living with HIV, 2.5 million new infections, and 1.7 million deaths in 2011. There have been over 250,000 HIV-related papers in PubMed. That's a far cry from the early years when very few people became interested in this disease, and in fact was the reason why even my own mentors tried to discourage me from making that dramatic turnaround in my career. But if you look at the investment and the science that has come out of that, it has been nothing short of breathtaking. From the very early studies about the epidemiology of the disease, the incremental increases in understanding some of the pathogenesis, and then finally the breakthrough of the etiology, the discovery of HIV, which led to the kinds of studies that have really transformed this disease and led to the interventions in the forms of diagnostics, therapeutics, and prevention. I show this slide, which is a very humbling slide for me because it really summarizes in one slide essentially everything that I've been doing over the past 30 years in HIV/AIDS, from studying primary infection, the establishment of infection in the lymphoid tissue, the massive viremia with wide dissemination to lymphoid organs, the very curious HIV-specific immune response that never completely controls viral replication, the immune activation, which I had been studying for years, well prior to HIV/AIDS, and this immune activation mediated by a number of factors, including cytokines, which is the engine that drives HIV replication. The accelerated replication, and in the absence of therapy, the destruction of the immune system. In 1993, 20 years ago, in a review in Science, I was again troubled and mentioned specifically my concern about the persistence of immune activation and said it may have a number of negative consequences. As the years went by, this became apparent because we know now, as a matter of fact, that this aberrant immune activation enhances HIV replication, it induces immune dysfunction by cellular exhaustion and other mechanisms, and it leads to chronic medical conditions such as the endothelial proliferation associated with cardiovascular disease, and in fact has given us insight into processes ranging from chronic brain disease to the entire process of aging in the human. Probably the greatest impact with regard to an intervention has been the basic science associated with studying the HIV replication cycle, from the initial binding, fusion, entry, reverse transcription, integration, transcription, and ultimately the budding of the mature virion. Because the study of these has shown that they are vulnerable targets for the types of drugs that have developed over the many years since drug development began, right after the recognition that HIV was the causative agent. That leads to the treatment of HIV-infected individual. Here we have me in the 1980s, this is the fall of 1981, making rounds on my patients, which really for me was the darkest years of my medical career and even my personal life. Because prior to this, as I mentioned to you, we had developed therapies that induced long remissions in inflammatory diseases. Now I was dealing with a situation where virtually all of my patients died. Here we have a median survival in the early 80s of 6 to 8 months, which means that 50% of your patients are dead in 6 to 8 months. But remembering what I said about therapy in the early years, in 1987, the first drug, AZT, gave us temporary optimism that we would have something that we could use to suppress the virus. But it was only with the development of multiple drugs and the combination therapies, which are essential for a highly replicating, highly mutating virus, that led us to have the combinations that we use today that have completely transformed the lives of HIV-infected individuals. So that if someone comes into our clinic in the same room where you saw me there in the early 1980s, and I started that individual on therapy and they were in the mid-20s, and they took their therapy, three drugs in combination, I could mathematically predict to them that they would live an additional 50 years. And a number of studies, including this recent one from Johns Hopkins, show the same thing, virtually normal lifespan. This is the developed world. The same is true in the developing world as long as you provide the drugs. This cohort analysis from Uganda shows the same thing, nearly normal life expectancy if you put people on appropriate drugs and bring their viral load to below detectable levels. I cannot talk about treatment or interventions without mentioning very briefly the role of activism in HIV science. I mentioned to you that I was the public face of HIV because President Reagan early on barely mentioned the disease, so when we were talking about it, I was the person in the newspapers, on television, and on the radio. And when the activists began frustrated by what they perceived as the slowness of the federal government in leading the effort against AIDS and the rigidity of the clinical trials process and even the regulatory process, they appropriately lashed out. And who they lashed out was me. And you could understand why. I was reading what they were saying and listening to them, and I began to realize that they made a lot of sense. Now, what they did is that they stormed the NIH, and for those of you who had the opportunity, and I recommend it, to see the Academy Award-nominated documentary, How to Survive a Plague, you might recognize some of these pictures of the activists demonstrating on the campus at the National Institutes of Health. Having read what they said and listened to them, I became convinced that in many respects they were correct. So I made one of the smartest moves that I've made in a long time. During that massive demonstration, I told our police officials to gather 5 of the leaders of the demonstration, to bring them to my office to begin what really turned out to be an ongoing dialogue to this day of how you get community involved appropriately in the design of clinical trials and in understanding the importance of regulatory flexibility. They were arguing for access to drugs that had not yet proven to be effective. You fast forward more than 10 years when we showed efficacy of the drugs, and in South Africa and all of Southern Africa, there was the same sort of activism. These individuals had borrowed a page from the playbook of the gay activists in the United States from the 1980s. And they were demanding access to drugs that we had already shown were life-saving. It was in this environment that another very important period of my life began. And that had to do with my involvement in the birth of the President's emergency plan for AIDS relief. So let me tell you briefly how it happened. President George W. Bush, who, as I mentioned, I had known from his father's administration, called upon me to go to Africa and design for him a program, if possible, that could bring drugs to the developing world, particularly Southern Africa. There were many people who felt you could not treat Africans because they'd not be able to take drugs every day the way we do in the United States. Was this correct or not? I went to Africa and I made the rounds in hospitals and in the bush. This is a very poignant picture for me. It almost brought me to tears when I was there, because here is an African doctor with a ward full of AIDS patients, and this was deja vu. This is exactly what I faced in the 80s. He had no drugs to give them, and essentially all his patients were dying. There were some organizations that were raising money trying to bring drugs to people. Here's one of those individuals from the AIDS Service Organization in Uganda. I visited her and went with her in the bush, and that case on the back of her motorcycle is full of drugs which she delivered with me to the villages in northern Uganda, in which these individuals here have a mother, if you look at her on the left-hand side, was infected, and she was taking care of her own children and the children who were orphans of AIDS-infected people there. And here she was taking her drugs regularly and was essentially getting back to normal. I put together, after several months, going back to the United States, working very closely with the White House staff who were extremely helpful, and put together a program which is now known as PEPFAR, presented it to President Bush at his request, and then in January 28th of 2003, he announced the $15 billion President's Emergency Plan for AIDS Relief, which is the most impactful global health initiative in the history of the United States. And together with the Global Fund, philanthropies such as the Bill and Melinda Gates Foundation, Médecins Sans Frontières, the Clinton Foundation, and importantly, the contributions of host countries, we have transformed the lives of individuals in the developing world. We have a long way to go, but if you look at 2002, when the president sent me to Africa, there were about 50,000 people in low and middle income countries receiving antiretrovirals. In 2011, the last time we have an accurate count, there were over 8 million people, which has now averted over 800,000 AIDS-related deaths. Prevention has been extraordinary. We realized early on that prevention is not unidimensional, but requires a combination of modalities. The low-tech ones on the lower level, testing, counseling, condoms, harm reduction. The biologically based ones on the next level, prevention of mother-to-child transmission, male circumcision, microbicides, treating STDs. But one that really stands out that I want to point out to you. When we were putting the PEPFAR program together, there was a tension between how much you put into prevention and how much you put into treatment. As it turns out, a landmark study has proven that treatment is actually part of prevention. And the study that I'm talking about was a more than 1,700 HIV discordant couple study. By discordant, we mean one is infected and the other is not. They're regular sexual partners. And it was shown dramatically that if you treat the infected person in that partnership and you bring down the viral load to below detectable level, you decrease by more than 95% the likelihood that that person will transmit the infection to their uninfected sexual partner. Now, I get back to this combination HIV prevention. Vaccines could be the nail in the coffin of HIV, despite the successes that we're already seeing in the absence of vaccine. There's a story about vaccine that is critical to the immunology community. Classic vaccinology, for example, the vaccines against diseases that maim and kill, like smallpox, measles, mumps, polio, they all do something that tell us that the immune system is capable of protecting ultimately even with a vaccine. They prove the concept because the majority of people recover and they're left with an immune response that protects them against reinfection. So vaccinologists have classically used the infection model as the paradigm for vaccine development. We have no such proof of concept with HIV. It is very, very different. The disease is almost invariably progressive in the absence of therapy. Interestingly, autologous neutralizing antibodies, namely to the autologous virus, appear curiously only after several months and yet they're still ineffective in controlling the viremia. Broadly reactive neutralizing antibodies curiously only appear in around 20% of individuals, and only after 1 to 2 years when they clearly are hypermutated and are a high enough affinity to actually broadly neutralize, but even then, they're not effective in completely controlling viremia. The virus is never completely cleared, and curiously, there's no protective immunity against superinfection of an already infected individuals. We've been searching for an HIV vaccine for over 26 years. The early attempts used the classic paradigm: get an envelope, immunize, get antibody, and hope you get protection. It did not happen. And so what we did is we called a summit in March of 2008 and published the results of that in July of 2008 in Science. And we said the way forward was to address the basic science and really get some answers from an immunological standpoint. However, we had previously started years before that an empiric trial, essentially, using a pox vector with inserts of HIV genes followed by a boost of a protein. And we had the first very modest signal of efficacy, but it was a surprise because this modest 31% efficacy signal was correlated with a surprising lack of neutralizing antibodies, although non-neutralizing antibodies were present, likely acting through antibody-dependent cellular cytotoxicity. There were no CD8 cytotoxic cells specific for the virus, and these antibodies were directed against an unexpected variable region of the HIV envelope. But moving ahead with the science-based approach, we took the clue that even though few people developed neutralizing antibodies, they did so, as a matter of fact, against certain epitopes on the envelope, such as the CD4 binding site, that we felt that if we could induce these antibodies, we could get them to actually be broadly neutralizing. And there have been some very elegant papers that have appeared over the years. The most recent one from Haynes and his group, people from the NIH and a variety of others, called Coevolution of a Broadly Neutralizing Antibody in the Founder Virus. In brief, these individuals had the opportunity of studying someone very soon after infection and looking over a 2-year period to the coevolution of the mutated virus and the hypermutated antibody. In other words, schematically, what they did was show that as the virus mutated, the unmutated ancestor antibody began a trek over 2 years until it finally resulted in broadly neutralizing antibody. Now, the big trick is how do you then induce that? And there's a variety of studies from a number of groups now looking at how you can target germline HIV immunogens to the germline B cells. In other words, how do you get an immunogen to bind to a germline B cell receptor to induce the kind of neutralizing antibodies that we need? This is a work in progress that's heavily weighted to immunologists. In other words, turning that neutralizing epitope into an immunogen. So if we could get that vaccine there, then the situation would really change. So take a look at where we are without vaccines. 22% decrease in new infections, 24% decrease in deaths. We had a meeting in Washington, D.C. in July of this year where we had the audacity to talk about turning the tide together. In other words, ending the AIDS pandemic. I had the privilege of giving the opening keynote address where I emphasized greatly the importance of the basic and the clinical research that would give us the interventions which, when implemented, could lead to the end of HIV/AIDS. We did modeling. You could look at either antiretrovirals, microbicides, prevention of mother-to-child transmission, male circumcision, and even a vaccine were we to have one. I show on this modeling slide, again, I made it simple for understanding, that even without a scaling up, we're seeing the kinds of improvements with treatment and prevention that I showed you on an earlier slide. If you scale it up a bit, you can make the deflection of the curve sharper. If you scale it up greatly, the deflection is even sharper. And if you get a vaccine and introduce a vaccine, the deflection would be even sharper. And so as Secretary Hillary Clinton said just a few months ago on World AIDS Day on December 1st, 2012, she unveiled what we call the PEPFAR blueprint. I had the privilege and the opportunity of working with her and her staff, particularly Ambassador Gooseby, who's the head of PEPFAR, in putting together the 4 roadmaps associated with this blueprint. And as you can see, there's saving lives by the combination prevention and treatment, which I spoke to you about, targeting the populations at greatest risk, having a global effort, but importantly, driving results with science, because science got us here, and I believe science is going to finish it. And unquestionably, the science of immunology is going to play a major role in getting us to what we will refer to as the first AIDS-free generation. And so once again, friends and colleagues, thank you for the opportunity of being able to present to you today. I wish you a very enjoyable and successful meeting. Thank you.